Hi everybody, welcome to lecture 2 of human biology. This is going to be a lecture on cells. Alright, my first comment on this is that it wasn't always completely obvious that living things were composed of cells. Now, being cellular is one of the main components of being living. So this is a very important part of science. It wasn't until the 1600s that we started to get microscope technology that was good enough for us to be able to see, to understand what cells are. It kind of started with some of the folks seen here on this slide. This is Robert Hooke, and this is Anton von Leeuwenhoek. They're some of the early practitioners of microscopic examination. This is a von Leeuwenhoek microscope. By today's standard, that's still a pretty good scope. What they were doing is viewing very small organisms in some cases, sometimes moving, sometimes not, and sort of doing this early work that helps us understand today what cells are and how cells function. In fact, it was Robert Hooke that coined the term cell when he looked at a wine cork many years ago, again 1600s, and saw the small little openings in it and thought it looked like cells, not prison cells, but the cells that monks slept in back in the day, these little hovels, little square hovels. So he actually coined the term. Now, a few hundred years later, Matthias Schleid and Theodor Schwann come along, and by the way, you don't have to remember the names necessarily. We're just looking at concepts here and kind of tiptoeing into the world of cells. It wasn't until a couple hundred years later, Matthias Schleid and Theodor Schwann came along and sort of defined what we now call cell theory. And cell theory states the following: all organisms are composed of cells and cell products. That means if you're alive, you got cells. Cells are the basic structural and functional unit of life. At its smallest level, cells begin what life is. So the smallest organisms on Earth are single-celled. Number 4, cellular activity reflects itself in the structural. Cellular activity reflects their structural components. In essence, this is getting at organelles. And last but not least, life is propagated through cellular activity. Okay, this sort of takes the magic out. Now, you don't have to memorize all this. My point to you is quite simply that we have not always known that we were composed of cells, and it hasn't been that long since we realized it. It's not uncommon in the 1800s. No one's washing their hands when they're doing surgery on somebody. Germ theory is not a thing until the mid to late 1800s. This is not that long ago in the grand scheme that we realized how all of our smallest bits are in fact put together. All right, here's a good old-fashioned generalized cell. I'm just going to throw some stuff at you real quick so you get an idea of what we're dealing with here. We've got the outside of the cell here, commonly referred to as a cell membrane. Inside of this cell, we have our good old-fashioned nucleus. The nucleus is where you house your DNA. We have rough endoplasmic reticulum with its ribosomes. We have Golgi body. We have some smooth ER over here. There's some mitochondria. All of these things should sound marginally familiar to you. Now we're going to go through everything bit by bit and learn all the parts and sort of what they all do. But this is a good old-fashioned cell as we know it today. This is specifically an animal cell, but we're going to look at how it fits together and how it is capable of doing what it does. Now, the first part that we're going to get into when we're dealing with this is going to be the cell membrane. Now, your cell membranes, the structure of them we consider to be a fluid mosaic, or referred to as the fluid mosaic model of cell membrane structure, a fluid mosaic. The idea here is that the outer surface of your cells, your cell membrane, is composed of what we call mostly phospholipids. Okay, this phospholipid bilayer, and it's going to have these polar heads on the outside and on the inside. There's some polar heads, there's polar heads. And then inside of here we have these nonpolar tails. So the inside nonpolar bit is going to be what we call hydrophobic, it's water-fearing, and the outer edge and the inner edge here and there are going to be considered what we call hydrophilic. They are water-loving. The take-home message to this is that your cell membranes, by having both hydrophilic and hydrophobic regions, they're going to be really selective on what they let through. Your cell membranes have a selective permeability. They have a capacity to pick what comes in and what does not. This gives your cells the ability to regulate its internal environment. So that's homeostasis on the most simplistic level. All right. So we have these cell membranes They're mostly made up of lipids, these hydrophilic and hydrophobic tails, these phospholipids, and a phospholipid 1,2 bilayer. Now, I will also point out the glycocalyx before I move any further. Studying along the exterior surface of your cells, let me say that one more time, the exterior surface of your cells, you find a lot of sugar molecules. Sugar, 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 sugar. They're everywhere, okay? This is referred to as the glycocalyx. Anything you see G-L-Y, glyco, think sugar, okay? Glycocalyx, this is a sugar coating on the exterior surface of your cells that's very important in maintaining what we call self-non-self recognition. In other words, you can think of the glycocalyx, the sugars on the outsides of your cells. Are there any images of it here? Not so much. But they would be studding all along the outside of this, okay? Just covering it, all these sugar molecules. Think about it as a barcode. Like you go to a grocery store, you're scanning things through the machine. The barcode says what it is. Your cells have a barcode that says where in the body they're supposed to be and that they belong to you. All right? It's really complex, much more complex than I'm making it here. But the idea is that your cells do indeed have a cellular barcode so that your white blood cells, the cells that defend you, have the ability to sense if that's one of yours or if it's not one of yours. In other words, a bacterium gets into your system, it doesn't have the right glycocalyx, so your body will attack and destroy it. This is how your immune system works. All right, so this is very important for cellular recognition, the glycocalyx. There are a variety of proteins that are going to be found within your cell membranes, and I mean a lot of different types. I'm just going to lay a few of them on you so you get an idea of what we're dealing with. So not only are your cell membranes going to be made of mostly phospholipids, but there will also be a lot of proteins. These can be integral proteins, being that they span the entire length, or they can be peripheral, being that they're found either on the inside surface or the outside surface. All right, so here are some integral proteins. These are going to span the entire length of the membrane. They go from the outside to the inside. Some of these, just going to be receptors, all right? Just generalized receptors. So we may have mentioned the effect of insulin on your blood sugar levels. When your body's blood sugar is too high, you release insulin. That brings your blood sugar levels back down. What insulin actually does is it moves around. It attaches to very specific very specific receptors on your cell surfaces and tells your cells to take up excess sugar, thus decreasing your general blood sugar level. These receptors are going to have a binding of some sort of hormone, typically on the outer surface, and then they will instigate a change inside the cell. Some of these can be enzymes that can help break things down. Some of these are ion channels or gated ion channels. Quite simplistically, ion channels let very small things through, like ions. They're ion channels. So hydrogen ions, chloride ions, they can just pass through and maintain a diffusive gradient. In other words, it can be at balance. Others of these are considered gated ion channels. Gated ion channels have the ability to open or close dependent upon the use of energy molecules like ATP. So ATP is your cellular energy molecule, stands for adenosine triphosphate. And if you bind an ATP molecule to one of these gated ion channels, it'll either open or close depending upon what it's made to do. Okay? For instance, if you are seeing me right now, that's because the rods and cones in your eyes are picking up light from the room around us and causing the opening and closing of gated ion channels to send messages to your brain about what you're seeing. In fact, that message traveling down a neuron to your brain, that's opening and closing of gated ion channels. Your ability to hear me right now is governed by very special what are called hair cells in the cochlea of your ears. And when vibrations pass over the hair cells, it bends them. And when they bend, they open gated ion channels, which signals the brain, in essence, that you're hearing certain things. So these gated ion channels are really important for us. Some of these can be cellular identity markers. Look at that. It's got a let's just keep this simple. It's got a sugar on its outer surface. This is a glycoprotein. It's a protein that bears a sugar, a glycoprotein. So this is going to be part of the glycocalyx that we talked about moments ago. And then others of these can be cellular adhesion molecules or CAMs. Cellular adhesion molecules are going to allow one cell to sort of connect to another cell to anchor itself in place. How deep do we want to take this? Not too deep. your white blood cells have an ability to move around inside your body. They don't just flow through your bloodstream. They can actually attach themselves to the walls of your capillaries and stay in place. They can get into your lymph nodes and stick in place and sit in your lymph nodes. The way that they do this is by using cellular adhesion molecules, these cans, like little hands, so they can grab a hold of and move around when necessary. That'll do. All right, other protein variations here. So what we have are tight junctions, desmosomes, and gap junctions. Okay, tight junctions, desmosomes, and gap junctions. Tight junctions are tight junctions. These are going to be areas where two cells abut one another, and the junction between them is held very tight, so nothing can get between the cells. All right. This is going to be found, for instance, in your intestinal tract, where we want anything that gets into your bloodstream from your intestine to have to make its way through the physical cells and be selected for. You may realize that you have a lot of bacteria in your gut. You don't want that bacteria getting into your bloodstream. So how do we prevent bacteria from getting into your bloodstream? We make the junctions between cells incredibly tight so nothing can get between cells. Again, the body has to select what gets through the cell into the bloodstream. Tight junctions are an important part of this. They form an impermeable passage between cells. All right, then there are desmosomes. All right, watch this. I can grab a hold of my skin here, and I can pull on it and not tear a big hunk of flesh off. One of the reasons for this is because your skin is just packed with desmosomes. What desmosomes do is they form these plaques, these little side pieces inside your cells that sort of connect one cell to the next. So one cell is going to connect to the next cell at the desmosome, but then the desmosomes have all these radiating fibers that go off in opposing directions. And by doing this, those fibers connect to other desmosomes, which connect to other cells and other desmosomes, and then other cells are connecting to other desmosomes, and before you know it, whole sections of the tissue are just packed with all of these intermediate filaments, these fibers, connecting with desmosomes, and it makes each cell no longer function as a single cell in terms of tension. It makes the whole tissue function as a single unit for strength. All right, so I can grab my skin and pull, and I can feel it getting tight all around my arm because my skin is linked to each other. All the cells of my skin, they're individual cells in there, but they're all linked to one another using desmosomes. All right, and then last but not least, we have gap junctions. Now, gap junctions are a little variation on the theme here. What they will do is have two cells that sit next to one another, and the gap junctions will attach those two cells to each other. The variation is that gap junctions have a gap, a little opening between the cells. What this will do is it will link cells together chemically. Here, we're linking cells together to prevent things from getting between them. Here, we link cells together based off of strength so that they can be strong together. Here, we are linking cells together chemically, so if there's a chemical reaction that happens in one cell, that chemical reaction is going to carry over and take place in the adjacent cell. Your heart is famous for doing this. You can stimulate the heart at one little spot at the top, and that stimulus runs through the entire length of the heart, stimulating the entirety of the heart almost simultaneously, because they are packed with gap junctions. Very important. And worthy of mention here, other cellular modifications to the outer surfaces of your cells. An important bit are macrovilli. No, not macro. Microvilli. Macro would mean large, micro means small. These are small, very, very small. Microvilli are important for forming undulating surfaces on some of your cells. Quite frankly, this is from the intestines. Very important in the intestines are these microvilli. What you want in the intestine is the ability to take up as much material, as much nutrient as is physically possible. You want the intestines to be incredibly good at pulling the important stuff out of the foods that you eat and getting it into the bloodstream. Well, how do you increase the absorptive capacity of a cell? You increase its surface area. How can you increase the surface area of a cell? You give it all of these undulating fleshy projections. These are microvilli. Microvilli are just finger-like projections that sit off the top of a cell. In this case, the top of a cell. They don't move, they're non-modal, they have no other major functions of that fashion. All they do is increase the surface area which increases the absorptive capacities of a cell where that would be found. Here's tight junctions, there's a desmosome, you can see some gap junctions, those are microvilli. Now, one of the things that people confuse microvilli with are cilia, and I think it's important to talk about cilia as a result of that. Cilia are little hairs. They're not big fleshy projections off the top of the cell, they're little hairs that sit off the top or the side or wherever on a cell. And these little hairs can be motile or non-motile. Okay? Sometimes cilia beat. They have what's called a power stroke and recovery stroke, a power stroke and a recovery stroke, and a power stroke. Okay? They beat and they move things from one place to the next. A really good example of motile cilia in this fashion is what's seen in your tracheal passages. Every now and again, while you're sitting there watching this video, you're going to go, and what's happening is you're breathing in all the dust in the world around you. You know, kind of dust your clothes and look in a light, and you'll see all sorts of stuff floating around in the air around you. That's English. And you're breathing that stuff in. Well, your respiratory passages have a lot of mucus on them, and they are going to get all this buildup of debris from the world around you that you're breathing. That debris gets moved up and pressed forward and up and up and up and out of your tracheal passages using motile cilia, where every now and again you go and you call that clearing your throat, right? What you're doing is you're clearing all that debris that the motile cilia moved up from your tracheal passages and swallowing it down into your stomach for digestion. That's how this works. There are also sensory non-motile cilia, and we'll talk about some of those as time progresses. In fact, the hair cells that I talked about in your ear moments ago, those are actually non-motile cilia. And when you bend them, it opens those gated ion channels. It all fits together, right? All right, worthy of mention here is the cytoskeleton within your cells. This cytoskeleton is incredibly important. It's going to be made up of three major parts that are worthy of our mention: the microfilaments, the intermediate filaments, and the microtubules. Now, your cells have a skeleton. Okay, just like you have a skeleton, your cells have a skeleton, and it functions in similar fashion. First are microfilaments. Microfilaments are important for general cellular movement. So the whole cell kind of has the ability to kind of move around a little bit in some cases by using these microfilaments. Your muscles are based off of this theory. In some ways they're microfilament based. Certain single-celled organisms that are capable of the whole cell kind of moving around, that's based off of microfilaments, so these are important. There are also intermediate filaments. Intermediate filaments are those which are connected to desmosomes. Now, desmosomes are important, as we talked about previously, using these intermediate filaments for strength. So our intermediate filaments, these are like ropes. Okay, they are flexible, but if you pull against them, they get very tight and go no further. All right, so the intermediate filaments are like the ropes of your cells. When I grab hold of my skin and pull, and it goes no further, that is the intermediate filaments becoming taut and not allowing me to tear pieces of flesh off. Okay, very important, these intermediate filaments. Again, associated with desmosomes. And then we have the microtubules. Now, microtubules are fascinating. These are like tent poles within a cell. They are rigid. They stick out and they stay there. They don't bend. They don't flex. They're like tent poles, okay? And they hold the cell open in its general shape. But microtubules have another very important function, and that important function is that microtubules, in association with what are called motor proteins, allow for intercellular movement. So if the cell has a vesicle or some sort of molecule that it needs to move from one place to another, it will actually attach to a microtubule and use it like a subway system and move across its surface to wherever it needs to go. Inside the cell, when you see something like, and bear with me folks, this and all this stuff is kind of in a spot, the reason for this is because there are microtubules that hold everything specifically where it needs to be. If this needs to move over here, it's going to attach to a microtubule and be carried over there. If that needs to stay right there, it's going to attach to a microtubule and stay right there. The microtubules form an internal motility network that allows things to get from one place to another. Membrane transport. All right, let me just say this. Your cells have the capacity to allow certain things to move and other things not to. But generally speaking, the smaller something is, the easier it moves. The larger something is, the harder it is to move it. Okay? Small things move easy, big things take a little bit more complication. And with that in mind, there are two forms of discussion that we need to have here. This is going to be on the subject of passive mechanisms like osmosis and diffusion, and then active mechanisms, passive and active. Active mechanisms like active transport, vesicular transport. The variation here is if it's a passive mechanism, it doesn't require energy, whereas if it's active transport, it's energy requiring. So you have to have an ATP to run this. This just happens naturally. All right, so first things first, let's talk about diffusion and osmosis. These are passive mechanisms. These happen without the use of ATP, all right, without the use of ATP. So diffusion is basically the movement of particles from high concentration to low concentration. If I had a jug of hot water here and I poured Kool-Aid into it, the Kool-Aid would permeate through the water and become one with it. That's diffusion, just the movement from high concentration to low concentration. I'm in a room right now with nobody else, but if this room had 500 kindergartners just stacked on top of one another, and I open that door, they would rush out into the hallway, just like diffusion, okay? Movement from high concentration to low concentration. And then there's osmosis, okay? Osmosis is the movement of water molecules from high concentration to low concentration. Typically, we think of this as being through a membrane. Now, the way we can look at this is by talking about what's referred to as tonicity, all right? Tonicity. If we take a red blood cell out of you, it looks something like this. Good old-fashioned natural osmotic levels. This is what your red blood cells look like. They're little pink donuts. But if I take that white blood cell and I drop it into hardcore distilled water, like just pure H2O, water is going to rush into that cell and make it swell up like a balloon. It might even pop. Alternatively, if I take that good old-fashioned red blood cell out of you and I drop it into hardcore salt water, like as salty gets, that salt water is going to pull the water out of the cell and make the cell shrivel up like a little bitty bag. And that's what happened here. Now check this out. Can you see the little spikes that are sticking off of this? Like tent poles? Those are microtubules. All right, you can see them here in this crenated or shriveled up cell. All right, those are examples of osmosis. That's water moving across a membrane. That's osmosis. Diffusion is movement of just particles in any medium. So if I took red blood cells and I dropped them into a big bucket of water, they would diffuse through it. That's diffusion. Osmosis is crossing that membrane. Movement across a membrane, the movement of water itself. I'm not terribly concerned with facilitated transport, although I will quickly mention active transport. Active transport, again, we're changing gears here. Diffusion and osmosis just happen naturally. They do not require energy, whereas active transport does. Okay, active transport is energy requiring. It has to have it. It is active transport. It requires ATP to function. And we're going to keep this kind of simple. Let's see, we have primary active transport, and what's going to happen here is we'll have a protein molecule, and that protein molecule is going to act like a pump. It'll grab a hold of something on the outside of the cell and pump it into the cell. Very simplistic. It's a pump, okay? To run a pump, like a pool pump or something like that, you got to have energy. This is energy requiring. It's just on a cellular level. So we have these little proteins that grab something and pump it into the cell. Grab something and pump it into the cell. That is active transport. We call this primary active transport. Endocytosis is going to be energy requiring. And this is going to be where the cell physically using energy will reach out with its cell membrane, will reach out and engulf a particle and bring that particle into itself, into the cell. Endocytosis, bringing things in. Exocytosis, taking something large and letting it out. Okay, so we have endocytosis, we have exocytosis. Endocytosis, bringing things in. Exocytosis, letting things out. The variation here is that primary active transport moves very small particles like individual ions, whereas vesicular transport, or endocytosis and exocytosis, this is for you moving very large things, like big old honking molecules, or large amounts of very small molecules. This is how you bring in large things, really big stuff, okay? A primary active transport molecule would only be about this big. We're trying to bring in big honking amounts of stuff, large amounts of things. All right, so let's go into the cytoplasm. What we're going to do is we're going to go through and talk about the individual, we call them organelles that are found here, and sort of what they do, and sort of everything that's inside the cell. And if we're going inside the cell, the first thing we should probably mention is the cytoplasm. Okay, cytoplasm is the fluid medium inside the cell. It's like the soup from which everything else is made and in which everything else sits. Okay, so cytoplasm is the fluid inside the cell. This is the fluid medium that everything else kind of floats in. All right. Next, we have our mitochondria. So mitochondria look like this here. There's a nice mitochondria down there. You may have heard these referred to as being the powerhouse of the cell. Not the way I like to think about it, but you know, it works. This is the site of cellular respiration, what we call aerobic cellular respiration. Mitochondria are important for making ATP. In your body, mitochondria make the ATP. One more time. You make ATP using mitochondria, which conduct a process called aerobic cellular respiration. Okay, very important here, very important. Mitochondria are pretty cool on that facet. Like you have cells in your body like your muscles that require lots of ATP, they have lots of mitochondria. You have other cells in your body that don't need a lot of energy, they have fewer mitochondria. So the number is actually quite plastic. They're cool. And sort of a side note, they have their own DNA. So mitochondria have their own unique DNA sequence because they're in essence derivative of bacteria. Mitochondria act like and look like interiorly little bacteria in your cells that do a job for you. It's like a symbiotic relationship, which is kind of neat. All right, let's go to the endomembrane system. We're getting a little technical here, folks. I'm just going to lay this on you in as simple a terminology as I can. I'm going to tell you how all this works. Pay attention. This is getting a little complicated. All right, here we go. What we have in the endomembrane system is a nucleus, a rough ER that's studded with ribosomes. We have a Golgi body, a variety of different vesicles, and then the outer surface of the cell referred to as the plasma membrane. All of this is a factory. It's just a factory. There's nothing fancy happening here. And it's a factory for making protein. Your cells are a factory for making protein. Okay? At the end of the day, that's what this is. What's inside the nucleus? Your DNA is. Your DNA will release a single-stranded copy of itself. Long story there, but that's what happens. That gets into the rough endoplasmic reticulum, where it will attach to ribosomes. Those ribosomes will make basic primary structures of protein that will then be offloaded from the rough ER to the Golgi body, where they're finalized, they're made in their functional forms, and those can either be released into the cell to do a job, or they can be released from the cell to do a job. So, blood sugar up too high, you need to release insulin. So what happens? Your body is, or this cell is signaled to make insulin, so inside the nucleus, the genetics necessary to make insulin are found. A copy is made and sent out to the rough ER, where ribosomes attach to that template, if you will, and make the primary state of insulin that is then sent to the Golgi body, where it's finalized and put into its usable form, that is then offloaded from the cell and released into the bloodstream to bring down your blood sugar levels. Your cells are a factory for making protein. Now, there's obviously a little more going on to this. There's smooth ER, which is very important for the synthesizing of cholesterol and steroid hormones. We've got lysosomes that are important for digestive processes in and of the cell. So there is some other stuff here, but knowing about ribosomes making proteins, knowing about the Golgi body finalizing things, that's the main part that I want you to be concerned with here. All right, here we have the nucleus. What I want you to know about the nucleus is very simple. This is going to be what houses your genetic material, and the nucleus is important because it has these little openings that are called nuclear pores, and they're going to come into play a little bit later on, okay? So just know that the nucleus houses your genetic material, and know that it's got little openings called nuclear pores that we're going to talk about at a later date, okay? Just remember that for the time being. Now, not all of your cells have nuclei. nuclei. Yeah, that'll work. And some of your cells actually have several. Okay, so generally speaking, like your red blood cells, for example, if we go back and look at those, and here, there's no nucleus there. That's what gives it this weird donut shape, is the nucleus has been pulled out of it. Your functioning red blood cells are devoid of nuclei, which is kind of crazy within the body. And then, by comparison, you've got some muscle cells, as an example, that have lots of nuclei. So most of your cells have one nucleus, some of your cells have lots of nuclei, and you have some cells that have no nuclei whatsoever. And I think that'll do for me. Now, let's talk a little bit about your genetic code inside of the nucleus, your DNA. So what this is, DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid, I'll be showing you this here in just a second. And inside of this, DNA itself is made up of a variety of nitrogenous bases. These nitrogenous bases are called adenine and guanine. The purines are adenine and guanine. These are the big ones, double ring structures. Double ring, purines, adenine and guanine, and then the smaller pyrimidines. The smaller pyrimidines are single ringed, and they are cytosine, thymine, and uracil. Cytosine, thymine, and uracil. Now, in DNA, we use adenine, guanine, cytosine, and thymine. In RNA, which is ribonucleic acid, which we'll be learning about here in just a second, it will use adenine, guanine, cytosine, and uracil. So thymine is in DNA, uracil is in RNA. And that takes us here. Now what we have, this is a general structure of a DNA molecule. Now in this, what we have is the good old-fashioned double helix. You're probably at least somewhat aware of the double helix, double helical structure of your genetic material. This double helix, what it actually is, if you unwind it, is an alternating covalently bonded sugar and phosphate backbone. So sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, an alternating covalently bound sugar phosphate backbone with these nitrogenous bases in the middle, being adenine, guanine, cytosine, and thymine in DNA. Now, I want you to know who binds to who for your exam. Adenine only binds to thymine, thymine only binds to adenine. Guanine only binds to cytosine, cytosine only binds to guanine. That is it. That is how this works. So, on your exam, if I were to put up a list of nucleotide bases, could you tell me what the corresponding opposing line would look like? What does adenine bind to? Thymine. What does guanine bind to? Cytosine. Adenine, thymine, guanine to cytosine, adenine, thymine. The way I think about this is, this straight looking letters like an A only bind to the straight looking letters like a T. And the curvy looking letters like cytosine, C, only bind to the curvy looking letters like guanine, G. Curvy letters and curvy letters, straight letters and straight letters. Adenine to thymine, guanine to cytosine, adenine to thymine, cytosine, guanine, thymine, adenine, adenine, thymine, guanine, cytosine. Just walk it down. And then if I showed you something like this one and that one, and I said, which of these is RNA? You'd say this one, because what does it have? Not thymine, but uracil. Okay, but uracil. And that's good enough for me. So we've got a covalent bonding set here. The central portion of this is hydrogen bound, and if it's hydrogen bound, these are weaker attractions. So if you're going to open up a DNA molecule, it's going to open up at the hydrogen bonds. That's pretty much good enough for me, folks. Let's jump on into the cellular life cycle a little bit. So how do cells reproduce themselves mitotically? Mitosis, right? That's where we're going with this. How do we do it? Well, the cellular life cycle is going to look something like this. We're going to have a phase of mitosis where the cell divides. That's going to be followed by a interphasic period. Interphase, mitosis. Interphase, mitosis. Interphase, mitosis. Interphase is where the cell is growing and doing what it needs to do as a cell, doing its job, and preparing for division. Mitosis is actually just a snap of the fingers by comparison. It's a very short time period where the cell physically divides. So for you to get larger, to grow like you have done in your life, you've got to have mitotic capacities. Your cells have to have gone through mitosis and increased in size and number for you to grow larger. But you got cells today that no longer go through mitosis. They are no longer mitotic. So let's talk about these just a little bit. What will happen is this. A cell goes through mitosis and then it enters this G1 phase. What happens is it grows larger and it reaches a functional size, and a lot of cells just stay here. The G1 phase of interphase, that's where the cell's doing its job. If you got nerve cells that are doing what nerve cells do, they're in the G1 phase of interphase. If you got liver cells that are doing what liver cells do, they're hanging out in the G1 phase of interphase. In fact, you got cells that live their entire duration just sitting around in G1, man. They're just hanging out there being cells. You got cells in your brain that have been in G1 for most of your life, and they're going to stay in G1 till the day you die. They are non-mitotic. G1 of interphase is where the cell does its job. If it ever goes into the S phase, it's going to divide. Because the S phase means synthesis. Okay? What you're doing is you're making new DNA, you're making new histone proteins. These are important for packaging your DNA material, your genetic material. The S phase is where you're getting ready for division. And then you got this G2 phase. The G2 phase is where you're really finalizing things for division. And then you hit mitosis. And when you hit mitosis, you get something like this. I will tell you the following. This interphasic period, all right, this is where the cell's kind of doing its job and getting ready for division. And then you have prophase, metaphase, anaphase, telophase. This is where the cell is dividing. And then cytokinesis is where the cell is finally divided. I will not ask you to name a phase of mitosis. I simply want you to know that mitosis is where the cell divides. I will potentially go into more detail on these phases of interphase because they just don't get enough love from my perspective in science.